Yeah, okay.
Good evening, everyone. Thank you for joining us in our last webinar with Joel. I am Jamili Zavala, member of the Western Canadian Soil Health and Grazing Conference Planning Team. I manage the Cara Soil Health Lab and then a Soil Health and Crop Management Specialist at the Chinook Applied Research Association. Please be aware that this webinar will be recorded. You will be muted. Feel free to write your name, where do you farm, where you are, what part of the world are you, and this is the last opportunity for your questions. But I warn you, if you don't have your question ready at the beginning, we are not gonna have it at the end. So put it at the beginning. Give us your opinion of this webinar series with Joel, and then tell us what you think about all of this good experience that we have. The Western Canadian Conference Soil Health and Grazing is a new movement relative to Western Canada, and the formal recognition began with the first Western Canadian Conference in 2015. And for the next conference in 2017, the planning team recognizing the connection with the forest, and then they joined it with a long-lasting grazing conference. That year was a sold out. And then 2 years later, in 2019, another sold out. The third biannual Western Canadian Conference supposed to be this year in December, but unfortunately, with all of this uncertainty due to what's happened with the COVID, has been delayed to 2022. To help keep the soil health movement strong, the conference planning team has been very happy to present this webinar series with one of the most popular speakers that we had from our last conference. Guess who he was? Joel. Joel is an independent plant and soil health educator and a strong soil health advocate. He studied agricultural science in Australia and uses an integrated approach in managing soil microbial ecology along with crop and soil nutrition to optimize plant immunity, soil function, and soil quality for sustainable farming. Now he's based in Ontario. Joel has worked intensively in Australia and in Europe, integrating chemical and biological soil assessment with plant nutritional analysis to better understand and manage crop production. We are very fortunate to have him, not only for a passion for sharing both the scientific and practical knowledge he has learned, but the ability to really connect with farmers. He has presented formal lectures to agricultural communities around the world. And we had the pleasure to have him for the last 3 weeks. The first week was talking about soil nutrients and then all plants nutrition and what was new in soil health, and a master class in plant nutrition. Excellent. On the second week, we have him talking about the transition to low input production system, where he gave strategies for, what we should be taken into consideration for transitioning in there. The last one was last week, where he was talking about integrating for regenerating strategies for regenerating forage stand and using perennial crop and annual forage in crop rotation. And today is our last week with Joel. Questions have been sent to us, and then I've been putting this part of this presentation that I have right now, and then we will go to this. But before that, I just want to get a little information, and looking back at the previous webinar, I just remembered some of this most important part of this literature review he put there. He put this one, maybe you remember about this meta-analysis where he was talking about how important is plant diversity. And something that caught my attention when he was talking about that is the gram positive to gram negative bacterial ratio decreases And then I tried to understand what it was that, and then I did a really good search online and which one were the gram-positive and which one was the gram-negative bacteria. And this come to me with more questions and say, oh my God, the gram-negative bacteria increases, probably this is something good because some of the gram-negative bacteria, so that kind of bacteria, that is one of those that fix nitrogen. So the article is right there. Here is the link. You can look at this and then you will have a lot of fun reading about that. The next one that called the attention that we talk about getting you some information was about the necromas. Very interesting information with the necromas. So right there, I found the link. You can copy this and you can look at it online and you will get more insight what the necromas contribution has to the soil organic matter. Another thing that was asked question there, one question that resonated to me, and especially because I have a little story to tell you about this question. Somebody asked about maturity. If there was any nutrient that could be substitute of the glyphosate for speeding crop maturity. I don't remember clear about if the question was about boron or they was saying something about, you know, if we apply a lot of boron, we can, speed the maturity. And I remember that I say something about, hey, no, probably it's all the toxicity and all that. And you all said that was a really short margin in there. But that reminds me something that happened in 2014 in a mystery piece tree that I'm going to share with you guys. This was something that we did in 2014. And then after all of this talk made me think about how important it is that the soil be in a good conditions. In this case, this experience was this. I don't know if you see right here, there's this peas are more mature. Here is flowering when the rest wasn't flowering. And I call that the mystery strip. The interesting thing is that I was planted on May 23. And we had a field day that day. And then Robin Bowman is a lady that is a expert in pulses that visiting. And she asked a question to me, when this was planted? And I say, May 23rd. And then she told me that was too late. That was planted too late. I feel so embarrassed that I wanted to disappear that day. But anyway, so I showed this one because you can see the strip right here is more green than the rest of the one. And then 2 months, less than 2 months later, it was very mature. And when you look at this in August 19, that strip matured 2 weeks before that time. What it was telling me was that something was happening in that specific area that allowed this to be 2 weeks ahead. Very quickly, I did some evaluation and then tried to look at soil physical, soil chemical, and then we didn't have the lab at that time, but this is what we found out. The strip that mature early, they have deeper soil for the compaction was more deeper in the soil. So there was more area for the root to grow. And then when I did the chemical evaluation, the most important thing to look at here was that that strip get more yield. And then what made the difference was the organic matter there. The organic matter was 1% higher than the strip that was, growing normal. So what happened there was that area was supposedly a feeding area, or was a fen along there, or what. So that's the story that I find out. But I wanted to share this because that reminds me that if we have a soil that has the condition for the plant to grow, we can get faster weight for the maturity is going to be faster. So that's the reason I want to share with that, which is nothing different what Christine Jones have been showing, around saying that when you have good soils, and then in this case, another fence line showing that the plant's growing better. So it's an indication that if we had good soil, we can increase the maturity, or we can get the maturity, when they're supposed to. This is what I want to share with you. Sorry it takes so long to me to get into this, but I want to share this. Then looking back, another question that was asked, that was there, that was asked to Joel that I think is very important to come back and put this question. So for you to refresh what Joel answered to this question. The question was, what is the best way to balance nutrients level, for example, high potassium and magnesium? The reason I pulled out this question is because most of the soils in our area have this issue. And then here is Joel's answer. First, balance your high minerals, in this case with calcium. If the calcium is too high in there, so in that case, we need to focus on increasing organic matter as an amendment, or you can use also complexing it with humic or fulvic as a short solution, and then in the long term, build up organic matter. Another thing, the strategy that he put there was monitor the crop. Why is that important? Is because when you have some balance of minerals in the soil, that is going to balance the nutrients in the plant. And so that's the reason he said, pay attention to that. And I just wanted to share with you guys this. And then this is pretty much a, little background information, what happened in the last 3 weeks. Now we're ready for the questions. So there was another question that was postponed for today. The question was, what were Joel, what were your thoughts about soil microbial carbon use, efficiency, or producer heating a ceiling on increasing soil organic matter? For example, they cannot get their soil organic matter higher than 5% despite following best management practices such as cover crop, no-till, manure, following all the principles. What is your thoughts about this, Joel?
Yes. Okay. Thank you for that introduction and a good recap and summary of where we've been the past month or so, for that last 3 weeks. So a good refresher for me as well. I can't always remember how I answered some of those questions. Yeah, this one was a tricky one. I read through this question and I kind of felt it would be really nice to know a little more context and really, I mean, I don't know if people can unmute themselves if they have permission in the settings of this call, but just on that note, I mean, if what's that?
If I figure it out, well,
yeah, I don't know if they do, but if the listener or if any of the listeners on these questions are in the room tonight, do feel free to unmute yourself and chime in and just contribute a little more detail or some. If you want to engage in some conversation, feel free. I'll just put the offer is there if you want to, and if they can.
Yes. So just if they send me a sign, and then probably I can look at this and then see if I can.
Yeah. That's a good idea. If people want to raise their hand down at the bottom, they could raise their hand. And if you want to chime in a little more and engage in some conversation, that'd be cool. There's no reason why not too many people in the room tonight. So, so this is a tricky one. And it's we're just not lovely to know a little bit more context and whatnot. If I were to start with just some of the general answers that we kind of did cover through the course of the last few weeks, I think I touched on this very briefly, and for those that have heard me speak before, they know that I talk about this all the time, but this importance of roots and not shoots, this importance that soil organic matter does come from root biomass, and as we particularly discussed in the first session, this emerging importance of the root exudates and those more labile fractions of carbon as well. So I think that straight away, I think that the strategies to think below ground, think about more of those roots is key. And one of the reasons that's really important is that actually in the last 50 odd plus years since the more intensification of agriculture, it is root biomass that has been the trade-off. We know that we've been increasing yields. Yields have increased, since the Green Revolution and whatnot. And okay, they might be stagnating somewhat now, but those yield increases those above ground yield increases came at a cost to below ground root biomass and below ground carbon partitioning. So I think consequently what is for sure is that if we went back and to look at some of the older varieties, I mean, heritage cereals, heritage wheats and traditional varieties, these kinds of things, einkorn and emmes and even spelts, they do, a lot of these older varieties, they do have much better rooting characteristics and below ground biomass allocation. And now the trade-off, of course, if the goal was to build soil organic matter and we grew more of those, that's all good. But the trade-off will be that your yield might be a little lower because, again, the plants are allocating that carbon below ground. So, but I don't necessarily see that as a huge problem. I think if you manage your inputs accordingly and lower your input expenditures, those bigger root systems, they will scavenge more nutrients from the soil. They'll scavenge more moisture and more nutrients from the soil. So you're going to leverage the existing fertility that you have already with those bigger root systems. So I think and to do that for example, maybe a few years, I don't know how long it'll take. Obviously, it's going to be different, but let's just hypothetically say maybe for 5 years or so, you could do that across 5 points in that rotation, build that root biomass, and then go back into a traditional variety and a higher yielding variety that by that stage you would have built up a really nice resilience and soil organic matter fraction. So I think one part of my answer, I would consider looking at some of the older varieties and even if not, just looking at varieties that have greater root biomass, that is greater root to shoot ratios. That's definitely going to be one of the big factors. And really linking to that, of course, is then the importance of that living roots principle. I mean, you mentioned you're doing all the soil health principles. I think I would really very strongly put the living roots principle at a front and center. We talked about that in the first session about this emerging paradigm of how important the root exudates and the more labile fractions are to grow because they more efficiently grow microbial biomass, and that biomass gets preserved as necromass, which is what the bulk, a good 50% of soil organic carbon is. So really what that practically means is that puts the living root soil health principle at a front and center. We've got to have living roots and living carbon inputs, those exudates and more fresh rhizole deposits. That's what's going to really help to drive microbial biomass. So I think consequently, again, I'd be putting that as a really core principle, really front and center is the living roots, maybe my priority, quite possibly. And then linking to that is that I also shared that study that showed it was a lab-based study where they were just a pot study where they were looking at feeding different quantities of root exudates, different qualities. So it was a high diverse mixture of exudates or a low diverse mixture of exudates. So the quality was different, but the quantity was the same. And they showed that greater diversity of exudates did induce greater microbial biomass response. So if we translate that practically, that means plant diversity. We want more species diversity. Therefore, we'll have more diversity of those exudates, and that potentially can be triggering greater microbial biomass, which then can potentially form that necromass. So again, I would think I would be coming back to the diversity principle, possibly really trying to focus a bit more heavily on, therefore, living roots and diverse living roots. And then lastly, just quickly, we talked about obviously plant nutrition. That was the very first session that we did. And I'll just emphasize that point that says, if you want to drive that photosynthetic factory, that photosynthetic machine, drive biomass production, be that above or below ground, etc. But in this instance, we're interested in doing it below ground. You've got to have adequate nutrition there to really drive that photosynthetic engine. So coming back to making sure that you've got adequate plant nutrition and therefore maybe doing things like foliars and that kind of thing to manage that.
Yeah, good. Our next question is related to management. This is someone that wants to do regenerative organic, is possible. So he will start with no-till seeding a cover crop mixture or annuals, year one. Then the question is, what can one do to set oneself up for success to get good plant growth without any of those inputs that has side effects?
Yeah. Okay. So if weed pressure was a concern, really, then I would say that your priority has to be to choose some fast growing annuals in that very first wave, in that first succession. Okay, actually, if we look to the evidence on, for example, cover crops and their ability to suppress weeds, and I can't remember if we discussed this in this webinar series or not, or if it was another one, but actually, because the key method to suppress weeds you want maximum biomass as quickly as possible to shade. And so in that context, actually, there's quite a few studies that suggest that actually a monoculture, which is the fastest growing, is actually the best strategy to quickly shade and cover and therefore suppress those weeds. So it might be that a mono is best, but you've mentioned that you've premised the question that you're looking to do diversity, and okay, there's other good reasons to do that as we've discussed. So consequently, though, I would then be choosing a handful of the fastest growing species and have a mixture of those. I think that if you're trying to get them established and keep the weeds out, that's got to be your ticket, fast growing and shading. And then, part of that other part of your question is, would weeds really be a problem? And yeah, I would argue that's a valid part of the question. I mean, maybe the sheep and your chickens might actually graze and have a feed on some of those weeds. Depends on what the weed is, obviously. But it is an important point. We have this, conquering view of nature, and it's there to be managed and controlled by us. But I think it is important to take a step back maybe and appreciate the value of weeds. They do have some value, obviously, just not when they're in excessive, but they have some value there. So yeah, I think maybe it is about being a little more relaxed about your weed population. Not having some of the weeds there is quite okay. But really the key is you've got to establish fast season, quick growing establishment. So really that depends on when you're talking, summer or winter. I mean, quick growing warm seasons might be your things like buckwheats, or they're very fast to grow. Millets and sorghum Sudan, things like that are very fast to establish. If it was cool seasons, things like oats are normally a good choice there. They're pretty quick to grow. And again, I quite like beans, or cereal rye would be another one, and even vetch and even brassicas. I mean, brassicas would be another good example. Brassicas and the mustard part of the family particularly, they're renowned for releasing a lot of those allelochemicals that can also suppress weed germination and weed establishment. So things like mustards, and again, they're pretty quick growing, plus they release some of those allelochemicals, a bit like the ryegrass as well. So yeah, I would be choosing amongst that kind of a list, depending on your warm or cool season when you're talking. The key is fast growing.
There's a question here. I don't think we didn't talk about too much about the Johnson-Su bioreactor, but the person want to know, is that really feasible to use that kind of, bioinoculants for a 22-acre property? And they said that this doesn't only work for a small plot like a garden. You have any?
Yeah, no, indeed. There's a lot of farmers, broadacre farmers, using the method. The interesting thing about the extract approach is that it depends on how you're applying it. So if you're using it as a seed treatment, especially, but also that liquid inject that I think we also talked about, actually you don't need huge quantities of those kind of extracts. So there's actually a lot of farmers using the Johnson-Su method on many thousands of acres. They might have multiple piles that they're producing. They'll use those kind of 1,000 liter shuttle tote containers, the steel cages, and kind of maybe have multiple ones of those, kind of 1 cubic meter each type thing. So yeah, there's many farmers broadacre using the method, and that's the beauty of the extracts. They go a long way. It might be 5 to 10 liters of extract per ton of seed, or they might be injecting, anywhere from 20 to maybe up to 50 odd liters per hectare, something like that. So yeah, it's very achievable on a large scale as well.
There's a question here that they want to know about tips for converting row crops fields to perennial pasture without using any photosynthetics.
Yeah, okay. I think in that instance, one of the best strategies would be to do kind of like an undersowing. So in that point of time, in your very final annual, your very final row crop that you're growing, before that comes to harvest, you'd want to do an undersow, maybe an interseed, or however you want to kind of do it. But that way, when you come through with the combine off that annual crop, you'll already have an understory there established. It might be small and, you know, obviously shaded out and small, but that's okay. It's there and it's established. And as soon as you let that light in from the canopy, once the crop is harvested, they'll be there and ready to go. So yeah, I think it would be some kind of an underseeded, interseeded type thing. Maybe it depends on where you are. I know broadcasting into standing crop doesn't work so well in the dry climate. So it depends on where you are, but it does work well in the more moister, humid climates. So even broadcasting and indeed broadcasting does, you know, one advantage of the broadcast is that you're scattering those seeds on across that surface area of the soil. You're not drilling in rows, so you can get better kind of mixed coverage across the area to again help with suppressing weeds and that kind of thing. So yeah, but I think the key is there. I would just try and get that, whatever it is you're going to grow as your next species, is your perennial, sorry, try and get it established, or even part of, you know, something established just to keep those weeds down, that then you can just roll into your perennial. So yeah, I would do something like that. Or obviously after harvest, just go straight back in and straight away and re-drill after harvest if you can't do the earliest interseeding.
Another question in management is, this is related to dry matter increase due to increased number of species. Said that this is definitely beneficial in grazing situation, and I want to know if there is any test being done what impact those species, if those species increases, will have in grain production. And then some comment says, will it only be affecting overall yield in a polycrop, or will it also be beneficial in a crop where one dominant species will be harvested and all the others will be understory? Mm-hmm. So what is the difference between these two systems? Is it
Yeah, okay. So yeah, in the last week's webinar, we talked heavily around plant species diversity, and I was predominantly leaning at and sharing grazing and grassland pasture-based studies, because that, yeah, we were doing more of a forage kind of theme for that webinar. When we bring this over to grain, it's more of a mixed bag of results. Yes, the principle is basically the same, and yes, there's a lot of the same beneficial interactions that can transpire in a cropping scenario as well. I would acknowledge the subtlety of that question, that definitely the opportunity for those kind of synergies and that enhancement is definitely more pronounced in forage, and it's just easier to design for in forage and grassland scenarios. And I'd say there's a lot more evidence to really support the principle in that setting. Now, in a cropping setting, yeah, it's no reason why not. You can also plant multiple plants together, and they're definitely, it can go either way. It can be competitive, it can be collaborative. And that's what's important about obviously designing that and bringing the right species and even varieties together. And that's an area of knowledge that there's plenty of anecdotal and farmer and field evidence out there, but there's a huge amount of knowledge gaps in that space. And I think, funnily enough, if we, even if you think about this whole idea of companion planting and this kind of thing, it's actually probably there's a lot more work been done in the world of gardening and vegetable gardening on that kind of scale. The idea of companion planting in vegetable gardening has been common forever, and so actually I'm sure there'd be all sorts of interesting learnings that could come out of going into that space and seeing how we could translate that over to agriculture. But nonetheless, so the potential is definitely there, both ways, for positive or negative interactions. And there are many examples of how it helps. I mean, the classic combination in a cropping scenario is to include a legume than with another non-legume companion. So the classic one might be with a cereal, but it could be an oilseed. But the legumes indeed do provide a great deal of benefit. They are particularly a special plant species to put into these mixes because obviously that they bring that free nitrogen, and that is a really significant contribution that they make towards helping the other plant. But also legumes are renowned for being very high, good mycorrhizal colonizers. They do have very strong associations with mycorrhizal fungi, and they do produce more acidic root exudates, a very unique set of root exudates that come from the legumes versus, I mean, I guess all plant species produce a unique set in their own various ways, but the fact that legumes are particularly acidic means that they're very good at solubilizing things like phols and particularly and calcium also. So those are the types of things that then can be shared with the other crops when you get that root intermingling. And that root intermingling is very important, that the roots have to be interconnected in order for these kind of nutrients to be shared and exchanged. So that would be one example. I mean, another one, we could go on and on, but I mean, another one would be grasses. They release a particular root exudate that's renowned for unlocking iron and zinc, something called a siderophore, a phytosiderophore. Microbes release these siderophores as well. And that's basically a chelating agent that grabs onto iron, particularly iron in alkaline soils and zinc, and makes them available. So the grasses are very good at solubilizing these trace elements, for example, and indeed then a legume or another companion with that grass or with that cereal can then also benefit from that. So those are the types of kind of interactions that we see where one species is very good at, activating a microbe in the soil or solubilizing a nutrient in the soil. And when those two root systems are intermingling, this is where we get this opportunity for kind of resource sharing. And really beyond two, yes, it keeps going. So you could have 3, you could have 4, you could have 5 crop species in there. The limit, there's no limit. The opportunities are endless. It just comes back to that practical reality of, okay, how do we manage such multiple species and that complexity of different species, different varieties, different growth tendencies, different maturity times, et cetera, nutritional requirements. I mean, it goes on and on. It just gets more and more complex, so complex. So that's it's really that's holding it back. But I think it depends what your outcome is for the grain. In a grain scenario, if you were combining it all and you weren't trying to separate all of those 5 or more mixtures, if you were just using it as a mixed grain feed for chickens or pigs and that kind of thing, then there's no problem. It's just when you move to some of the other separation, harvestability, maturity times, harvestability, separability, all those kinds of things, it gets a lot more complex in that cropping scenario. So, but nonetheless, if we come back to the core interaction and the core principle, the ecological principles there, then yes, there are these mechanisms that equally work in row crops, whereby nutrients, different plants can have these different niche complementarities. One might be deeper root, one might be shallow rooted, and then this resource sharing is the example that I just gave. So,
wow.
And then that last part of your question is yes, for sure. So if you were doing one main cash crop and all of the one other or more or multiple others were all, I would call that a companion crop. So you're still only taking one, it's not really an intercrop, you're only taking one through to harvest, but you've got all those other plants as auxiliary plants or companion plants. Then yes, I would say that there's a ton of benefits still to having those companion plants, even though you're not going to harvest them. And again, legumes would be a good example. For example, having understory of clovers would be a good example. I see there's a lot of work with micro clovers, little leaf clovers, and very small prostrate kind of varieties who aren't so upright and aggressive, but very low-lying clovers, for example. They are more of a companion for example, for the cereal. So yes, then you get a lot of those same interactions in the root systems, but it does make the practical agronomy, the harvestability, all of that kind of thing, it makes all of that so much easier. So I think there's a lot of opportunity for companion planting as well as intercropping, but companion has a certain ease about it, which is also still valid for those root-to-root interactions.
It's good. They're going to have more roots into the system. That's the part that is the most important in here. I'm going to try something here. We have one attendee that wants to ask some question live. Let's see how it works. Kim, are you able to talk?
I sure can. Can you hear me?
Yes.
Ken.
Oh, great, great. And greetings from the southwest corner of Victoria. I'm here with Helen, and we have very much enjoyed the seminars that Joel has put on. I took in a webinar a few months back that the East Gippsland Land Care Group put together, Joel. So that was the first time I heard you, and you're doing a great job in terms of, you know, putting out some good, solid, clear science behind your comments. I've got a lot of stuff on here, and greetings to Brenda as well. And I think I heard Ken Ziegler as well. I think he's there also somewhere. Anyways, greetings to you all. So we have a beautiful little place in Rocky Mountain House called Four Clover Ranch, and the Grey-Wooded Forest Association has done a lot of really neat work with us up there. Back in 2006, probably have to go back a little bit more. Grant Lastuka did some overwintered forage stuff, and we saw a huge transition into species composition. And Albert actually in 2006 took some species compositions of one particular paddock, and then we have 24 species there. So it really has changed dramatically in a very positive way. So then that paddock was part of a soil benchmark study that the Forest Association also did, and we just got the results, so it's very, very timely. And we were excited to see that our soil biology rated at 100, got a mark of 100, but our chemical, soil chemistry was not so good. It was down to about a chemical health of 44. But we just feel that we're going along very well. We've reached an equilibrium in terms of production and species. If we were to dabble into, perhaps some ripping or even trying some multi-species cover crops in there to free up some locked up phosphorus, we would change too much of a good thing that we've got going there. So I'm just really curious to hear Joel's comments on that, because as we then jump to Australia, where we bought our farm in 2013, and it's been converted from cropping with raised beds, very, very heavy volcanic clay soils. And we want to do sort of the Four Clover Ranch method and just changing the grazing management. And we're just struggling big time. So we've done a lot of cover cropping, and it's sort of shown some results, but it's just too slow for our liking. So we're thinking of liming. We've got some very high magnesium and iron levels, very, very heavy clay pan underneath, not very far down. Our tillage radish, and they develop T roots. A lot of people know of J roots, but ours are very special because they develop T roots, so they just can't get through. And one interesting thing, a part of the farm was put into perennial pasture right away when we bought it, and that has shown an increase in pH from 5.4 to 6. So I'm curious to hear from Joel if that management has been the contributing factor to that raise in soil pH. We think we're going to jump the gun and lime the rest of it, but it's just real challenging. So I'll stop and look forward to hearing Joel's comments.
I'm muting you.
Awesome. Okay. Thanks, Ken. Yeah, there was lots there. So I think one of the important things that you said, let's start with over here in the Rockies. One of the things you mentioned there is that although your soil biology was good, you were perhaps a little concerned that the chemistry result wasn't quite so good. But you also said that the production was good, that the plants seemed healthy, and that you seemed to be happy with production. There didn't seem to be any major issues in the plants themselves. And I think that to me is ultimately the overall master variable, the overall indicator. It's plants that we're trying to grow at the end of the day, and if they tell you that, you know, they're functional, they're productive, you're happy with the level of productivity out of them, and they're doing the job for you, then okay, a soil test might tell you it's this or that, but at the end, that's at the end of the day, that's a number on a piece of paper, isn't it? So the plants are the ultimate and master variable and goal. So I think if in that sense, if the forages are working and the pasture production is working, then obviously you're doing something right, and something is working. So I might not get too hung up about the chemistry test. And also just to say that, you know, I mean, I promote testing. I'm a big use of testers and both plant and soil analysis and that kind of thing. And I do see it as an important tool. But yeah, I wouldn't want to be, you've always got to ground truth it. You know, you've always got to calibrate what you see on paper to what's happening in the field. So, and don't forget that, you know, sometimes the chemistry tests, the way in which we measure the minerals is maybe not necessarily the best way. It comes from an era with a very somewhat blinkered and soil chemistry focus. You know, we're moving out of this soil chemistry view of the soils, encompassing more importance of biology and physics along with that, thinking more holistically. So, you know, along those lines, soils, you know, the tests that we do are often more of those plant available fractions. And I think I talked about this, you know, you would have heard me talk about this. So although maybe some of your plant available fractions are a little bit low, potentially hence your downgraded score, I'm sure you'd have adequate and quite good amounts of the total nutrients. So, and that you're not necessarily picking up with the traditional soil chemistry tests. So those will be there. And the main way in which those are going to be unlocked and cycled is through biology. So if you had a good biology score, then to me, I think that's, that and the good pastures is the ultimately says to me that things are going in the right direction. And so then to your other one in Australia, that sounds a bit more trickier. Yeah, clay soils. Yeah, classic high mag clay soils, they can be notoriously heavy and difficult and tight and difficult to work with. You mentioned iron as well. Iron can also lead to like a chemical compaction in a very tight soil. So yeah, you mentioned tea roots. I've not heard of that before, but I can certainly imagine it. It sounds like yeah, I can imagine that in such a soil. So I think I would say that yeah, I'm not opposed to liming. I do think that people can get into a bad habit of over liming, putting too much on. And I think it's when we're moving up to these crazy amounts like 10 tons to the hectare and these kind of things, it just sounds, it's ridiculously too high. I think I'm not opposed to it as with smaller amounts, you know, maybe 1 or 2 tons in that kind of ballpark, 2 at the max really, to the hectare. You know, and do it a little bit slowly and not necessarily to shock the system. But the key to lime as well is not just the lime itself. I think you've really got to have, again, that biological cycling of it. So I think having, again, might be diverse pastures or might be the use of those cover crops or those strategies to kind of then get roots and root exudates and the associated microbes to kind of work on that and assimilate that and turn it, you know, turn that calcium into their bodies, you know, transfer it into their bodies so that you get more of this biological functioning, I think is also really important. So I'm not opposed to a bit of lime. I think and that links to your question, do you have to use lime? No. Can management change soil pHs? Yes, I do think so. And I think that your example that you've quoted is one of that. I think with better grazing management and that you can change those things. And that comes down to with better grazing management or with more diverse plant stands, you can get deeper roots down. I'm sure your soils would have lots of calcium in them. It's just that high magnesium is obviously exerting a greater negative pressure. But if you could unlock some of that calcium that's being antagonized there and bring it up into the biological form, bring it up into the grasses and be recycled back through the animals and the manure and that kind of thing, then yes, that will be changing the soil's chemical characteristics. And I'm sure that's partly the response of, as to why you saw that in your other example from 5.6 to 6. So I think then just to address finally that compaction, I think that we often talk about tap roots for this purpose, and I think you really need a combination actually of tap roots and deep-rooted grasses with their more kind of fibrous and fine roots. You really need the action of both the tap and the fibrous fine, and together they can then punch through some of those heavy compacted kind of zones. Tap roots on their own, although we often talk about them being great for punching holes and opening up the soil, and they can, they're not so good at getting into some of that really tight stuff. You need very fine roots to start the process, and then that taproot can kind of follow through and really punch its way through. So I think definitely I'd be looking at deeper rooted species of grasses, you know, your timothies or cocksfoots, or I mean, any of your other deeper rooted natives there, of course, and then some taproots.
Yeah, that's a good point, because we also have a lot of issue with compaction here. So you're giving a good information for can be applied to our conditions too. I asked Connie that raised his hand, but we're going to go to a next question that was done before, and then we go live with Connie.
Cool. Yep, sounds good.
Okay, which one? Okay, the next question would be, and this is also related to, this is related to alfalfa hay, alfalfa. They said that in a hay field, how does one keep growing condition favorable for alfalfa? It looks like 6-8 years, the legumes start disappearing. And then they say that probably is because of the fungal bacterial ratio. So they want to look for something that keeps the production of the alfalfa. And then it's just, you know, asking question, should we chuck this with some fertilizer, or that will, you know, affect the bacteria? Or that will have some effect in setback nitrogen fixation of the alfalfa. So
yeah, okay, I'll be honest and say I don't have a particularly good answer for this one. I have heard of alfalfa stands just growing tired, and that they do often need reseeding, and I wasn't sure if maybe that could be something to do with some of our genetics, you know, some of the varieties, modern varieties that we've bred, that have become a little lazy and you know, dependent on being fed and inputs and that kind of thing. So whether or not there was maybe some link with some of the more modern breeds, I wasn't sure. But I think for me, so I don't have a clear answer on exactly how best to help that, apart from I think that I would suggest, however, just going with a little bit more diversity in that hayfield. I would try and include a bit of another grass. I know that's another common combination would be, again, something like timothy and alfalfa together. I just think that, and maybe then a few more steps beyond that with a couple of extra species. But I just think then you'll get a bit more of a diversity of roots, and then again more different actions and functions into that soil. And it's not just that one monocultural root system, one monocultural exudate pattern. You know, having some grasses in there, they're going to be a bit more fibrous roots. That's going to help to, again, provide a bit more bulk and organic matter, maybe stop a bit of that compaction when you're going through and doing the cuts and that kind of thing. So I think I would probably just say that I would try and turn it into a little bit more of a diverse hayfield and introduce maybe some longer term perenniality through that. Now, if however the alfalfa still begins to die out, or certain species begin to die out over time, I agree. I wouldn't want to dig it all up either. I think I would just try and really explore opportunities for just kind of stitching in, kind of overseeding and just direct drilling as a kind of a rejuvenation of the pastures. I wouldn't necessarily work it up. I would just try and kind of, stitch some new seeds in and get them to establish. Okay, maybe that might mean tickling the soil surface or a very mild disturbance there, just to kind of cut some grooves and cut some surface area to help those plants establish. So maybe a little bit of a mild and shallow disturbance just to assist that process. But I agree. Yeah, I wouldn't want to rip everything up and start again. I would try and do some kind of overseeding and. But okay, again, to do that, obviously you couldn't choose very slow growing and very finicky plant species. They're not going to compete. You know, you'd need to be overseeding with some varieties and some species that do have a bit of vigor in them anyway. They're gonna compete against what's already there and establish a bit better. So I've not come across it to do with the fungal bacterial. Could be. I've not come across that at all. I guess maybe. What are you suggesting there? Maybe the fungal bacteria declines, so it's going more bacterial, and then therefore that being a factor. Could be.
Yeah, but also could be something related to some diseases that the crown of the alfalfa could have, because you know I've seen some alfalfa plantations that have that. You don't even see that until you dig into the crown to see what the issue is. So probably
that's a good point. Yeah, that's a good point. And I don't know if I shared it this time in the presentation, but there's plenty of good work that shows that as you increase plant species diversity, actually the disease pressure declines. So that could well be it. Yeah.
I have Connie Maxon, the one that raised his hand. He wanted to ask a question. I'm going to allow him to talk. See.
Oh,
are you there?
She said she's raised her hand in error. Maybe she doesn't want to.
Oh, okay. So let me go to, well, Keen wants to talk again. We need to be careful with Keen, please. I'm going to allow you to talk. Keen?
Sorry, I'll be very quick. I put that in a chat. We actually built a Johnson Su bioreactor a year ago, and it'll be ready here in a month or so. But we're looking at ways to get the fungal dominance up in our soils. So asking Joel if there are other land management options for increasing the fungal component in our soil biology.
Sure. Yeah, I think that's, you know, the Johnson Su method does produce a good fungal compost. So I think you are barking up the right tree already. I think that's probably, that would have been one of the things I would have suggested. I think the other obvious things, like obviously try and minimize your soil disturbance and that kind of thing. You could look to other organic amendments. Your scale might be too big and they might be too unaffordable, but you know, looking at trying to produce a compost for spreading organic amendments and, you know, wood chip based kind of composts can grow good levels of fungal there, but that might not be too relevant on your scale. But I think the other last thing I would suggest is that there's also very good evidence that shows that, again, as you increase plant species diversity, you do encourage more fungi, both saprophytic and mycorrhizal fungi. They both respond to greater plant species diversity. So I think I would definitely optimize that, and then use something like the Johnson Su as an extract because again, you don't need large amounts. It's just an inoculant. You're just trying to put that little bit of diversity there, and then you'll let the diverse plant stands feed and multiply that small inoculant. Let them do the work out there in the field. So yeah, I would either again use Johnson Su as a seed treatment to do if you were doing any reseeding, or I would just kind of knife that in and inject that into an existing pasture, and just kind of cut that in and dribble that down into the soil. To apply that, or dribbled into the furrow if it was a cropping scenario.
Let's keep going with this question. Here is a question about nutrient management. Instead of fertilizer, what crop can we interseed to provide phosphate?
Yep, buckwheat would be the classic example there. Buckwheat and legumes. I mean, any of the legumes, again, they release those acidic exudates, so they're very good at solubilizing phos, and buckwheat is very famous for that. So I would do something like buckwheat or any of the legumes.
If sulfur is needed, what do we see cover crop, or we buy inputs?
Yes, either, maybe a combination of both. You know, so I would say in terms of cover crops that are good sulfur accumulators, again, it's the Brassica family. They're rich in those sulfur-bearing compounds. So I would use something like any of the Brassicas. maybe even the forage radish. I know we often talk about the radish being a good nitrogen accumulator and that deep root and pulling up nitrogen from depths. Well, sulfur is also a leachable element. So, and the radish is also a brassica. So it's also a good accumulator of sulfur and can pick up leached sulfur at depth as well. So, so yeah, I would think about a brassica or any brassica, or particularly the forage radish type thing. For scavenging sulfur. And then, okay, if you needed to apply it, yeah, sulfur, it's tricky. Sulfur is very leachable. So it does have a tendency to be deficient in a lot of soils. But I would just say that a lot of the sulfur is also bound up in the soil organic matter. So if you've got good soil organic matter, you will have a lot of sulfur embedded within that organic matter, which is a store there, which you can kind of cycle and utilize as well. So if you've got good organic matter reserves, you also may not need to be applying sulfur, but if you're on a very light sandy soil and have low organic matter, then yeah, you're probably going to need some inputs. Yeah.
What crops are high demands potassium?
Alfalfa again would be one that's has quite a high, and any of your kind of hay crops generally remove quite a bit of K, as I'm sure many of you would know. I mean, and then beyond that, it's a lot of the vegetables, like fruits, I should say. I mean, tomatoes and cucumbers and citrus and you name it, anything fruiting. Potassium is really important for all fruiting crops. So anything that's fruiting, but beyond that, some of your hay and alfalfa would be other examples.
This is a question of how do we change pH if needed?
Depends if you need to increase the pH or decrease the pH. If you're trying to increase the soil pH, you would use things like liming materials. That could be calcium lime, dolomite lime, gypsum, these kinds of things. If you were trying to decrease the soil pH, you would use normally things like elemental sulfur would be what you would use. But really, I would say that, yeah, your goal maybe isn't necessarily to change the pH. Your goal is to maybe optimize the balance of those major cations, look at the kind of the balance there and just try and optimize that balance. And when you do that, you'll find that the pH will be somewhat, will be kind of self-correcting. It will fall into the natural spot. But also don't forget to say that plants themselves, I mean, we've had that heavy focus on pH, but plants themselves, they do release all those root exudates and they do change the pH in the rhizosphere. And the rhizosphere pH and the rhizosphere soil conditions can be completely different from and bear no reflection on the bulk soil pH or the bulk soil nutrient availability, for example. So that's where, when we then move to things like talking about plant species diversity and a greater diversity of roots and exudates and all that root intermingling, well, under those conditions, you are creating a micro environment, a microclimate in that rhizosphere that behaves completely differently to the rest of the bulk soil. So the plants can change the pH in that zone easily by 2 orders of magnitude. So that all becomes easier with more plant species diversity, in which then they can change that and therefore still be accessing nutrients and things from the soil within that very different microenvironment.
What is a rule of thumb for a sulfur to nitrogen ratio for cereals? Maybe for protein production, probably.
Yeah, so it's normally about 1 to 10, 10 nitrogen to one sulfur is commonly what's required by the plant, and so often people would kind of apply it in that ratio.
Which form of sulfur is preferred in a liquid status?
Okay, so the form of sulfur that's plant available is sulfate. That's the form that the plants actually take up. Now, elemental sulfur can also be used, but strictly speaking, it's not available as elemental sulfur. It has to be oxidized or exposed to that oxygen where it will then form sulfate and then can be taken up. But that said, elemental sulfur can have a tendency also to volatilize off as sulfur dioxide, a sulfur gas. And indeed, providing you've got a living plant, it's not on applying that onto bare soil, but if you've got a living plant, some of that sulfur dioxide, that sulfur gas that comes off can indeed be taken up by the stomata. And so the plants can use sulfur gas as well for a source. You can get small amounts through the stomata in that way, but the bulk of it really has to be oxidized into the sulfate form, where it can then be taken up. Yeah, I mean, again, you could use granular or liquid. You can also do it through foliars. You know, any of these are options. It's just a matter of what you're set up, what's practical, and whichever way you're going to do it. It's you know, again, always follow some of these rules like combine with carbon and that kind of things that we discussed.
Here, if someone has issue with cadmium in their soils, so any advice to reduce this cadmium?
Sure, it's yeah, cadmium, I'm pretty sure, is normally more available under more acid conditions. So possibly, depending on what their pH is, but possibly a liming material to increase the pH will reduce the availability of that cadmium. So that would be one. You're not getting rid of it per se, but you're just trying to basically make it not bioavailable and not available, therefore not to be taken up. So probably increase the soil pH, increase soil organic matter. I think I've seen some interesting work with earthworms and worm casts having some interesting abilities to really very strongly bind and complex heavy metals and immobilize them. So whether or not there's scope to do something around with encouraging more worms or the use of worm composts to kind of help to bind, immobilize. Yeah.
Two questions here about soil and sampling analysis and chemical analysis, about where in Canada can be done that SAP analysis that you mentioned through one of the webinars. And the other one is about sampling, the soil sampling related to where they can do this analysis for the whole. I think they are talking about is the total,
yes,
total minerals in the soil.
Yes, that's right. Yeah, that's what it looks like. Yeah, okay. There is, so most people, the SAP analysis has become very popular through a company in the Netherlands who have kind of pioneered the approach. So you actually be surprised. I mean, they're incredibly proficient and efficient. Farmers from Canada and I know US and even Australia, all around the world, send them samples, and they are very quick to return those. But actually there is a lab that has just recently opened here in Canada called Future Analytics. I could paste that into, I know the website, I can't remember the girl's name. She's in Red Deer actually, but there we go, I just pasted it there into the chat. But, so that's the only lab specifically in Canada. There's also a few other labs that do it in the US as well, but that I'm aware of anyway, that's that one, the Future Analytics is the only one that, that here in Canada that does SAP analysis.
And for the total analysis, I will say we could do at the lab, but we don't have an ICP. We have the way to digest the sample to get the total, but we don't have an ICP. So maybe they needed to send it to a commercial lab that do the total.
Yeah. Yeah, I think you find there's quite a few labs that can do it. You just have to ask them for that totals test. Yeah.
They need to ask for wet digestion with nitroperchloric acid or sulfuric acid, and then that's the way to get out. Okay, there's a question about microbes here. Does the leader or lignin still needed to control microbes to lock in nutrients balance?
Yeah, for sure. Yeah, I talked heavily about the importance of the more available fractions, the labile, and how that's better, more efficient at feeding biology, and they can grow more microbial biomass with this higher carbon use efficiency material. We had all that kind of conversation, and that's all part of the emerging paradigm. But your question is still valid, of course. Yeah, litters, complex litters and lignans, they are still very important for a whole host of other reasons. Like I think I mentioned, you know, soil protection and moisture conservation and erosion protection. insect habitat, earthworm food. I mean, there's a ton of really important reasons why those complex materials are still really important. And yes, lignin does behave as your premise of your question there. Lignin does behave as a brake. It's that balance between carbon and nitrogen that either means that it will be released quickly or slowly. So more complex lignin component there does therefore by default mean more of a slow release, and that there can be certain advantages to that, not necessarily triggering the microbes into a crazy feeding frenzy. So there can be certain balances to that, and definitely the lignin helps with all those other functionalities. However, as I say, part of the new paradigm really is, which is really pinpointing on this carbon use efficiency, which basically simply refers to how much of per unit of carbon that we feed a microbe, how much biomass do they put on, how much weight do they put on, and how much do they respire off as CO2. And the higher the efficiency, the more biomass they grow and the less they respire. And so all of the evidence is highlighting that these more labile fractions indeed do grow more microbial biomass and lead to less respiration, and hence this new paradigm. So now I think that all that said, I think the practical take-home message, I guess, is really to say that it's not necessarily one or the other. In a lot of dryland climates, again, I know your lignin and your residue-based stubbles are super important for moisture conservation. Same deal. I know farmers in Western Canada, and it's the same in Australia. Many of them don't like growing legumes and don't like growing pulses in the rotation because they find it does stimulate a bit of breakdown of the surface mulches, which is a big problem in a dry climate. So I think that practically what this means is that the traditional view has been very focused on no soil disturbance and maximizing residue. And I think that's been the paradigm of let's build soil carbon, let's manage for soil carbon, has been stop tilling and keep soil armor. Okay, and I think there's nothing wrong with that. We don't necessarily have to change that. What we just have to do is bring in this additional piece of the puzzle, which says, and so do both of those, and manage for living plants, and manage for living roots, and manage for those root exudates. I think that's the practical, it's not to abandon either of those two critical pieces of the puzzle that got us here. It's just to say that we need to integrate the role and functionality of green plants, of living roots and those root exudates, and bring them both together.
So we have a lot of questions here. Do we ever gonna finish those, or where are we on timing here?
Yeah, 10 past.
Okay, so there is some question about amendments related to humic acid. Let me see. Okay. So adding carbon as a fertilizer, I couldn't understand what the meaning of this is. Was 5 to 8 liters for what? For meter? For humic acid? I don't have.
Oh yeah. No, I understand this one. Yeah. So I was talking about adding a liquid carbon source to the fertilizer granules. And yes, that is correct. So I was suggesting around about 5 up to 8 liters of liquid carbon source, and it could be humic acid or fulvic or fish or seaweeds. Yes, that is right. 5 to 8 liters per ton of fertilizer granule. And then in terms of storability, I think that the take-home message is that providing it's dry, then the granule has time to dry, then no, the flowability is okay. I think, but the take-home message is I wouldn't do it I wouldn't risk it and do it too far in advance. If you want to do all your coating, I would be doing it close to drilling season anyway.
We lost you. You are not clear. We lost you, Joel.
storage, but a short-term storage, like, you know, I can
Your
still hear you.
internet is,
Okay. Yes.
your
Okay.
internet, okay.
How much? Can
Sorry.
you hear me?
Yeah,
okay.
that's better. Your internet was giving issue.
Yeah, annoying. Yeah, so I was saying, I was just saying there that I wouldn't, after coating the fertilizer granule, I wouldn't put it into long-term storage. No, I would still do it fresh as best. I would do it as close to drilling as you can, but a short-term storage would be fine, like a week or so. That kind of thing would be okay, but I wouldn't put it into a long-term storage. Do it as close as you can to drilling. Yeah.
Another question about amendments. What are your thoughts on fermented applications?
Yeah, I watch these with interest. I see this gaining a lot of traction recently, the Korean natural farming and these bioferments. I see quite a few farmers doing this. I think it's in a way, it's in its early stages. It seems to be, I know a few farmers in Australia doing this as well. Some have invested quite heavily on brewing tanks and doing all of this kind of stuff, and have seen some very promising, good early results. So I think it makes sense. I think we had a kind of whole school of thought that was very anti-anaerobic, you know, that whole kind of school of thought there that was disease-causing organisms are anaerobes. It's, you know, there are both beneficial and detrimental anaerobic organisms. So the fermentative types, like lactic acid bacteria, etc., you know, in our guts, all the bugs in the rumen, and etc. You know, I think there are many beneficial anaerobic organisms, and indeed, also in soils, we always talk about soils being aerobic environments and the importance of aeration, and of course it is, but that is, you know, through the pores and that through that porosity. But within then aggregates, soil aggregates, actually they are lower oxygen environments, and so we want oxygen through the porosity of the soil, but then within some of those aggregates is actually, we want to occlude some oxygen. That's what helps to preserve some of our soil organic carbon and prevent it from oxidizing. So within that context, soil is a mixture of aerobic and anaerobic microenvironments. Overall, yes, it's aerobic and we want air, but I think there's a lot of opportunity amongst some of the anaerobic style microbes and some of the benefits they can bring. So yeah, I watch with interest. I think there's some interesting potential there for sure.
There is question about amendments, in this case, amino acids. There are a few of this question is related to kind of like a fish hydrolysate. As far as I understood, for the same reason, one will consider them in foliar applications. What kind of rates will one be looking at?
Yes. Okay. Yes, you can use it. Amino acids work very good through the foliars to combine with nutrients, and they help to chelate and improve the uptake of nutrients. But they also do the same thing to things like glyphosate or other ag chem as well. Now, if you were using something like fish hydrolysate, I could say yes, the application rate would probably be somewhere around about 2 to 3 liters per hectare. However, you can get some commercial amino acid products that are a very specific mix of maybe just one or some amino acids. I mean, glycine is a very common one because it's one of the smallest amino acids. So it's very commonly used as a chelator for foliars because it's small and it leads to rapid uptake. So if you were using a product like that, a commercial product, then I would say I would follow the label rates because it might be different if it's just a concentrated amino acid. But if you were using something like a hydrolysate, protein hydrolysate generally, then it's going to be in that kind of realm of 2 to 3 liters, you know, per hectare for a foliar. And if it was a soil, it could be 5, even as much as 10 liters if economically you can afford it to per hectare of soil for a soil application.
Yeah.
And okay, yeah, well, like delivery method, yeah, you can do either. That's
your internet is giving us a little issue right now. Hello? Hello? You're gone. Sorry. Well, Joel, your internet is getting.
Yeah, just dropped out there. How do I sound
Sorry, guys.
now?
Apologies. Hello, are you there?
How do I sound now? Yes, can you hear now? Okay. Yeah.
Joel, it's hard to. Okay. Joel,
Okay.
we don't hear you pretty well.
Yeah, can you hear me now?
And now we can hear you, yes.
Sorry, I'm not sure what's happening there. I can hear you. I can hear you totally fine.
Sorry.
Yeah,
I
I'm sorry.
think you partially answered this next question that is here. What kind of application does a foliar? And then let's see if we can go into, there is only a question about enzymes. So this say in a holistic management organic cropping system where foliar nutrition, biological inoculants, and biostimulants are being utilized, will there be a benefit in applying an enzyme product to improve the efficiency of biochemical reactions in the system over and above what the soil biology are already producing? So what are your thoughts about using enzymes? There are several questions about enzymes.
Yes. Okay. Yeah, good question and tricky one to answer. Mike, it really depends. I mean, the microbiology in your soil is going to be producing a ton of different enzymes already. All of the, okay, you mentioned you're already using inoculants, and then of course then your native organisms. All of those various organisms are going to be producing a whole host of enzymes. So if you know you're using these words like you're already doing a lot of things, you're doing foliar nutrition, you're doing inoculants, you're doing biostimulants, it sounds like you're doing a lot already. So you've probably got good microbial functionality and therefore probably a good enzymatic kind of release and processes. So you may not need specific ones, but part of that question then really depends on, well, what is your goal? What are you trying to achieve? So you know the second part of your question there. Put it this way, is your goal to liberate phosphorus? Well, then I would say you would be better to apply specific phosphatases, those enzymes that will liberate that phos, compared to a mixture, compared to applying a mixture of enzymes. You know, you're applying a very targeted one to do a very targeted job. Do I want to decay litter? Well, you'd be better to specifically apply cellulase or lacase and some of the other enzymes that specifically attack your litter. So what's your goal? And probably if you're looking for a very specific function, probably a very specific enzyme would do that job better than a mixture of enzymes. But if you're looking for general soil functionality, general soil performance, etc., then yeah, maybe a mixture would be better in that sense. But again, your biology is going to be doing a lot of that for you. So yeah. you wouldn't be needing too much of these.
Yeah, there is more question about enzymes, but I want to see plant hormone probably apply the same thought that you just share, right?
Say that again, sorry.
Well, say there is another question about plant hormone, probably will be the same effect.
Yes, correct. That's right. Yeah, there's another question about hormones, and that's a really big topic actually. It's a tricky one to just quick answer that in a quick question, but yeah, you're right. So hormones can have very specific functions, and they can be complementary, or they can be they work in kind of opposites. So if your goal is a very specific function out of that hormone, you want to, for example, you're particularly trying to encourage rooting, root biomass, for example. Well, okay, auxins will specifically help to do that for you, and that's whereas cytokinins, for example, they're generally a bit more shoot promoting. So focusing on one or the other, you can then steer in the direction that you want. Whereas applying the two together, yeah, you get maybe more of a counterbalance kind of thing. So yeah, similar with hormones. Hormones is a big topic actually. You had a few questions there, and it's yeah, it's I think, you even coming down to like seaweeds and kelp extracts, they're a good general tonic in that sense, but even different species of kelps can have different balances and different ratios of those hormones, so it can affect things in different ways. So yeah, it's not a very straightforward answer to that one, I know, but I think it is consequently very difficult to in a broad acre setting, it's very difficult to be so precise with your timings. Under a broad acre setting, it's, sometimes you just, when you can get out there is when you can get out there, and especially in northern Canada, northern prairies, the crops, they grow fast, and so it's I think it might be a bit much to ask to do it on a broad acre scale. I think in a horticultural setting maybe, but in a broad acre scale it'd be much more difficult.
Yeah, I have one last question in all the questions that were sent to me before today. I don't think we're going to have the chance to go over all the questions that was posted today here. And then probably this question has no answer when you read about this, because it's hard for doing research so detailed like this one that was asking. This is about root exudates. Are there any scientific paper about it which can provide information in more details about total or daily amount, difference in plant growth stages, and difference between species, and so.
Yes. There are, not to every question. So I would say this, root exudates are a very difficult part of plant sciences to study. They're notoriously difficult to study. When root exudates are exuded from the root system, they can often only migrate, 1 millimeter or so, and it might take literally seconds before they're consumed by a microbe. So they're only going to migrate literally a few millimeters into the soil. And if you've got a hotspot of microbial activity, which the rhizosphere is, you've got millions of microbes there, they're going to digest those root exudates so fast. So consequently, pretty much almost all of what we know about root exudates comes from lab studies, hydroponic studies, solution culture studies. There's only a very small number of in-field and applied root exudates. And there's a very good paper on that, which is called Root Exudates Mission Impossible. And it outlines some of these kind of technical challenges to studying root exudates. So it's an emerging area of this plant sciences. There's lots of really good work happening in this space. There's a few studies that have collected root exudates from in-field, so it's kind of coming. But there is bits of the answers to your questions, but I wouldn't say there's a comprehensive single study. What I would suggest is, and I'll paste a link here into the chat, but people might be familiar with Google Scholar. You can search scientific papers through this search engine. You could use try Google Scholar to search for papers. Now, Google Scholar is not the best search engine for academic papers, but it's free and everybody has access, and you can get, a lot of open source papers. So some aren't, but at least you can read the abstracts. But so for example, if I paste in this search command, I'll just paste that in there. People can click on that. What I've pre-programmed that command to do is to take you through to a result that says root exudates and then a question mark. So you could use, for example, that link to just replace the question mark with, what do you want to combine your search term with root exudates? What do you want to know? Root exudates and plant species, type that in, or root exudates and growth stages, type that in. You can change the question mark to whatever you desire. So you can use things like Google Scholar. As I say, it's not the best, but I mean, it's better than nothing. And by grouping terms like, and putting root exudates, as you can see I did there in inverted commas, that means the search term is going to search for those two words combined, root exudates combined, as one term. So it's going to look for root exudates and, okay, you might want to type in wheat, you might want to type in disease suppression, you might want to type in soil carbon sequestration. I mean, you could type in anything. Okay, so Google Scholar is, if people are looking for scientific papers, that's a reasonable place to start. And of course, you could type in anything else that you want to search for. But all I would say is then to answer the question, yes, different plant species do release different cocktails of exudates. Yes, we are beginning to kind of map those and understand that. Yes, we do know that plants definitely release the bulk of their exudates in the vegetative stages and very little in the reproductive stages. So you will get the most of your root exudations in that first 4 to 6, maybe up to 8 weeks, in that kind of window. It's the bulk of it is in the vegetative stages.
Well, there is a question here. Say, when are you back in Alberta, and how is Mrs. making out? And that is from,
I'm not sure. That's not for me.
that's not for you.
No, that was a question for Kim, I think.
Well, but I can answer that question. When are you coming back to Alberta? It is probably, we want you here, even will be for the Canadian Western Conference in Soil Health and Grazing, or you're coming to the Cara Soil Health mini conference in OEN. Just
Yes.
wait for the conditions to get better. So
Yes.
I don't think we're going to go through all the other questions that were posted today, and then I'm going to try to copy them and see what we can put together into the sense. And then I just want to, in the name of the Western Canadian Soil Health Conference Grazing Conference Planning Team, we would like to express our thanks to you, Joel, for this series of webinar that you've given to us and everyone. It was so wonderful, and there was a lot of good information. And then I also want to congratulate all of those attendees that were able to start planting new seeds with all of this new information in their heads for them to start, making a little changes in their operation. And not only that, they need to start nurturing that with a lot of diversity, microbiology, and then a lot of those, diversities in plants, and taking in consideration all of these nutrients management that they need to do in the brain first to start working into their field. Well, thank you, Joel. It was a pleasure to have you, and then we're looking forward to continue collaborating with similar webinars.
Absolutely. Yeah. Thank you very much for having me. I've really enjoyed the series too. And thanks everybody for your questions and discussion and feedback and comments. It's been also very good. And yeah, indeed, of course, I can't wait to be back in Alberta. I hope to be there sooner than later, that's for sure. And hopefully we can all go back to a sense of normality soon. So that sounds good. I'd love to come again. Yeah.
Good. Goodbye, everyone. Thank you for being part of this last webinar, and then we'll see you around. Thank you.
Thank you.
New message. See what is a new message? It's 2 new messages. Come on. Thanks to both Joel and Jamili, you both great teachers. Okay. People are leaving. I'm going.
Okay.
Bye, Joel. Thank you.
Okay, cool.
I'm
Thanks for having me.